This is part 2 of the blood lecture. We briefly discussed leukocytes, but let's talk a little more about their form and function. So leukocytes are the least abundant formed element in blood. They should account for the least amount of your blood volume, and you only have a lot of them if you're actively sick. So a normal, healthy person who does not have an active infection should have a tiny amount of leukocytes. The function of these things is to protect against infection, fight pathogens, and all immune responses. They should spend just a few hours in the bloodstream, using that as a transport mechanism, and then they should go to the site of infection. So your body makes these white blood cells, they're released into the bloodstream, and then from the bloodstream, they go to the actual site of infection. So if you cut your finger and you had some dirt get in there, there's bacteria, pathogens, there's always a risk for infection. So your body is going to release white blood cells into the blood, and then they are going to travel to the connective tissue in the finger and eventually make their way to the infection site or the injury site. They need to get to the area so they can eat any bacteria that is in the area to prevent it from getting in the blood and spreading to the entire body or going systemic. So leukocytes look different from erythrocytes. Remember we talked about erythrocytes dropping all of their organelles at maturity so they can carry oxygen. They have hemoglobin and they carry oxygen. So they don't need a mitochondria, they don't need a nucleus, they don't need any of those things. They just need the hemoglobin. White blood cells are a little bit different in the fact that they have a nucleus and they retain their organelles for protein synthesis. So if proteins need to be created, the leukocytes are going to retain the organelles that are responsible for that. So it's going to retain its nucleus because it needs its DNA and RNA. It's going to retain its ribosomes. It's going to retain its rough ER. And it's going to retain its Golgi. All of those things are involved in protein synthesis. There are also granules in the cytoplasm of a white blood cell. So when you stain them and look at them under the microscope, you can see these little granules that look like grains of rice. Those are obvious in white blood cells. All white blood cells also have lysosomes. And if you remember back to cells from Anatomy 1, lysosomes are the waste-eating organelles of a cell. So they eat bacteria, they eat debris, they eat anything that's dead or junk that we don't want in the cell anymore. And it's really important that leukocytes have a lot of these because their job is to get rid of waste, bacteria, pathogens, and things like that. Granulocytes, so the white blood cells that have these grains, have specific granules that contain enzymes that help fight against pathogens. So these grains have things in them that also have immune response and defense properties. So they're not just there for the look. They actually have enzymes that are really good at helping fight certain things. And depending on the type of white blood cell, there are different enzymes in them. So some leukocytes have grains or granules and some do not. The ones that do have them are called granulocytes, as in cells with grains, right? So all of these are made during myeloid hematopoiesis or myeloid leukopoiesis. Okay, so they're made in the bone marrow. These are your phils, all of your neutrophils, eosinophils, and basophils. They're just called the phils. Okay, they come from the bone marrow and they all have granules in them. These granules are the things that contain those immune enzymes, right? The cells that do not have granulocytes or grains are the agranulocytes, and these are the ones that are made in the lymphatic tissues. Okay, so these are your monocytes and your lymphocytes. So the ones that are not phils don't have grains, and they are made in the lymph tissue and not in the bone marrow. So what is the importance of the different types of white blood cells? Why do we have both granulocytes and agranulocytes? What's the difference between the neutrophils, the basophils? Well, depending on the infection or the issue that your body is fighting, you're going to use different types of white blood cells. Okay, so neutrophils are the most common in bacterial infections. So if you get a cut and bacteria gets in and it gets infected, you're going to expect to see an increased number of neutrophils in your body because their job is to fight bacterial infections. Okay. They phagocytize bacteria, meaning they eat it, destroy it, and they release antimicrobial chemicals that also help and fight that bacteria from spreading. If you have things like chickenpox, sinusitis, which is like a runny nose and congestion, or diabetes, you might see an increase in basophils. These secrete histamine, which is like a allergy response, and this helps speed up blood flow to the injured area. And then they also secrete heparin, which promotes the mobility of other white blood cells in the area. So heparin keeps the blood from clotting, keeps it thin. Speeding up blood flow and keeping the blood thin allows the white blood cells to travel easier to the area that they need to go. So basophils are for chickenpox, allergy type things, diabetes, and they make blood flow better to the area so more white blood cells can easily get there. And when you have an infection, you don't always just release one type of white blood cell. You might release 2 or 3 different types depending on what's going on in your body. So if basophils are released and they thin the blood, it helps other white blood cells get to the area as well, not just basophils. So eosinophils, they fluctuate greatly from day to night, seasonally, and with phases of like the menstrual cycle. They really increase in parasitic infections. So eosinophils are like white blood cells that help when you have like earthly or dirt related like infections or things going on. So worms, parasites, worms, allergies, so anything outside, collagen diseases, and diseases of the spleen and central nervous system also cause you to have an increased number of eosinophils. They eat the antigen antibody complexes, so things that are going to cause reactions in your body. They eat allergens, and they release inflammatory chemicals. So if you're having allergies flare up really bad because it's pollen season, the eosinophils are the things that are giving you the inflammatory response. They release enzymes that weaken or destroy parasites such as worms. So like tapeworms, intestinal worms, those kind of things. These are the white blood cells that attack those. So all those fills are your granulocytes, or the ones made in the bone marrow. These are your agranulocytes, or your lymphatic made white blood cells. So first we have lymphocytes, and their name basically tells you that they're made in the lymph tissue, right? Lymphocytes. The numbers for these increase in diverse infections and immune responses. So they don't increase just because you're sick. There's usually something much like more deep going on, like a stronger infection or a complication in the body that is really, really complex. So things like cancer. You'll see an increased number of lymphocytes when somebody has cancer. They put antigens on bad cells, so they flag bad cells as, hey, this is bad, we need to destroy it. And then they also coordinate with other white blood cells or other immune cells to help destroy that thing. They secrete antibodies and they provide immune memory. So let's say you get polio or chickenpox or some crazy virus, measles or mumps. These are the things that are going to flag that as bad and try and coordinate with the rest of the body to fight it. And then they're also going to secrete the antibodies. So next time this thing presents itself, your body recognizes it as bad much faster and it can fight it much quicker. So they play a role in immunity and immune memory, in becoming immune to things with like when you get vaccinations, your lymphocytes help play a role in creating the immune response for those two. So your body remembers how to fight something after it's like healed from it. Monocytes, the numbers increase in viral infections. Okay, I remember this because mono, the kissing disease, it's a viral infection. It's highly contagious. Monocytes fight mono. So mono is for viral infections. So if you get a virus, a stomach virus, a respiratory virus, you're going to see an increased number in monocytes. They don't work the same as all the other ones because viral infections and bacterial infections are entirely different. So they have different types of pathogens. Viruses mutate. They just act differently in general. So we need a different type of cell to kill it. So they leave the bloodstream and they turn into macrophages, which are cell-eating cells, right? They eat other cells. And then they phagocytize or eat and break down pathogens and any debris that are from that. And they can also act as an antigen presenting cell, meaning they mark the virus as bad so other cells can attack it as well. So there are cells in your body that mark things as bad and say, hey, this is bad. attack it anytime you see it. So monocytes act as that too. We talked about red blood cells and how they had a 120-day lifespan if they make it their full life. White blood cells also do not live forever, and they do not always stay in your bloodstream. So granulocytes leave in 8 hours and live 5 days longer. So they leave the blood within 8 hours of being released into the blood, and then they only live for about 5 days. Monocytes leave in 20 hours, and they transform into macrophages, and they live for several years. So you can keep monocytes in your body as a viral immunity, and it will easily attack the virus next time you get it. So that's how you can get a vaccine for like a viral infection, and your body will respond to it quicker. So like a flu shot. You can get the flu shot, and then when you get the flu, your body identifies it quicker because you still have those monocytes retained from getting that shot. And it easily identifies the flu as a virus and it attacks it quicker. Lymphocytes provide long-term immunity over decades. So these are the things for like measles, mumps, rubella, polio, those things. If you get those or if you get vaccinated against those, the lymphocytes are going to hold that long-term immunity for decades or even lifetimes because they're continuously recycled from the blood to the tissue fluid to the lymph and back to the blood. So they are moved back and forth, and they don't get destroyed or wasted or anything like that. So you can draw your labs and you can check to see if you have lymphocytes against certain diseases. Disorders in leukocytes. So we know leukocytes are the immunity cells. If you have a leukocyte disorder, you're probably struggling to fight infection. So you probably get sick really easily. So when you have not enough white blood cells, so anything below 5,000 is called leukopenia. So penia means really small, and leuko means white, right? So a really small amount of white blood cells. These can be caused by radiation, poisons, and infectious disease. And then the risks that are with this, so if you have this, you are at risk for infection. So it just makes your immune system weaker and weaker. Usually, people get leukopenia commonly from chemotherapy because chemo is a poison technically, so it completely diminishes the immune system and wipes out all the white blood cells. Leukocytosis. This is when you have too many. So high above 10,000, it's usually present if you have infections, an autoimmune issue, severe allergies, or some kind of disease that makes your immune system go crazy. Your white blood cell count will identify which type of white blood cells are really, really high, and then they can try and narrow it down from there with what is going on. If it's allergies, you might have high eosinophils. If it is a raging infection, we should expect to see high neutrophils. So they can figure out what's causing it. Sometimes it's an infection. We can, treat it and get it better, and your white blood cell count will come back down. Sometimes it's an autoimmune issue that you can't really treat. You just have to kind of slow the immune system down so it stops attacking itself. But these are both disorders of leukocytes. So leukemia also is a leukocyte disorder, but it is cancer of the tissue that makes the white blood cells. And it's usually the bone marrow. Myeloid leukemia is cancer of the bone marrow, and it causes uncontrolled granulocyte production. There's lymphoid leukemia, and it causes uncontrolled lymphocyte or monocyte production. Acute leukemia is sudden. It just happens really fast. And usually people die within months of it because it's so fast. It's hard to treat and stop. And then chronic leukemia is one of the slower growing leukemias that it's usually not detected for a while. And usually people can live for a couple years with it. You can treat it to an extent. It's not as rapid as acute, but it still does destroy the immune system. So the reason why this is a problem is because when it's uncontrolled production of these white blood cells, they're not being produced properly. So it can cause the immune system to have no working cells or very little working cells, or it can cause your blood to have way too many white blood cells in it. And then you have the problems with the osmolarity and the viscosity. The cell percentages get all mixed up. You have too many white, not enough red. It just causes a whole bunch of problems when the numbers are out of whack. And that's leukemia. When you get labs to check your blood count, this is what it is. It's a complete blood count. And what it tells us is that it's the hematocrit, your hemoglobin, the total count for your red, your white blood cells, and your platelets. The differential, so how many blood cells do you have of red compared to white? How many neutrophils to basophils to eosinophils? Those things. And then it also tells you the size of your red blood cells, and then the hemoglobin concentration per red blood cell. So does every red blood cell have 4 hemoglobin sites, or they only have 2 or 3? So just one lab test can show us all of these things. All right, so moving on to the clotting part of blood. So we all know that blood has to clot at some point. At some points it clots unnecessarily and causes problems, right? So the things that cause clotting, both good and bad, are platelets, and they're called thrombocytes. They are small fragments of big cells. So they're like little cell chunks, and they stick together and they cause clots and plugs to stop bleeding. Your normal platelet count should be between 130 to 400,000. Any less than that, you're going to have clotting, like problems with clotting. Any more than that, you're going to have excess clotting. So platelet function. So we know platelets are the clotting property of blood, right? The thing that clots. But what else do they do, and how do they make these clots happen? So first, when you have an injury to an area, the platelets are going to sense the injury, and they're going to release vasoconstrictors that help constrict the blood vessels. So this will hopefully shrink the blood vessels down so less blood can flow through them, and it prevents blood loss. After that, the platelets are going to stick to the injured area and then stick to each other. Okay, so they easily will stick to injuries and to each other to seal up small openings. This should create a temporary plug in the hole or the injury, wherever you're bleeding from. Okay, they're going to secrete procoagulants, which are clotting factors. And this is going to promote clotting of blood in that area. So anything that is called a coagulant or that coagulates is a clotting thing. So procoagulants is all for clotting. They're going to release those to get clots happening. This is going to initiate the formation of clot-dissolving enzymes. Okay, these are going to chemically attract neutrophils and monocytes to the site of inflammation or injury. So anytime you get hurt or cut, you're going to have inflammation in the area. So the platelets are going to send out these chemical messengers that call the neutrophils and the monocytes to come to that area, because we need neutrophils for bacteria and we need monocytes for any kind of viral infection that may have entered, right? They're going to get there and they're going to destroy and eat anything that they see in the area that is not good. Any bacteria, any dirt they got in there, they're going to eat it. Okay, and then the platelets are going to release growth factors that stimulate mitosis to repair the blood vessels. So they're going to release these growth factors that make the blood vessels copy their cells to repair the wall and repair the tissue in the area so there is no cut or injury anymore. Okay. After this is all fixed, the clot will dissolve away. That's what that clot dissolving enzyme is for. So that'll wait for the repair and then it'll dissolve away. So that's like a scab, and as time goes on, the scab will, eventually break off, and it's because it's dissolving away. So when something is bleeding and it stops, it's called hemostasis. So that means just cessation of bleeding. Okay, so platelets play a huge role in stopping bleeding. They can stop potentially fatal leaks. The smallest cut can become fatal if it just continues to bleed over time, right? So platelets plugging that, stopping the bleeding is huge. When there is a massive amount of bleeding, platelets usually don't help much with that because it's too much at one time to fix. But that is called a hemorrhage, excess bleeding. So during hemostasis, there are 3 mechanisms. First, there's a vascular spasm, then a platelet plug formation, and then your blood clots. And platelets are important in every one of these steps. You're cooking in your kitchen. You cut into your finger. What happens? So the body's like, oh, we got cut. We need to stop the bleeding. The first thing that's going to happen is a vascular spasm, or the vessels are going to constrict in the area. Okay? Less blood flow to the area, less blood loss to the cut. So this gives your body time to start initiating the clot process, right? So we're going to slow down the bleeding, give us time to initiate those clotting pathways. There are sensory receptors and stuff in the injured areas that will send signals to the brain that will cause the vessels to constrict. And then also, like I just stated before, the platelets release vasoconstrictors that also make the vessels constrict on their own. So there are nerves that sense everything, and the platelets also do that. The second step of hemostasis is the platelet plug formation. So you have your vascular spasm and your vessels are constricted. Now we're going to start plugging the cut area. Okay, so a healthy uncut vessel has a nice coating inside of it called the endothelium. So it's the lining inside the vessel, and it's coated with this slick substance called prostacyclin. That has a platelet repellent in it. So platelets do not like to stick to that. Okay, when a vessel gets injured and the endothelium is cut or ruptured, there is no prostacyclin in that area. So platelets will stick to the cut area. That's why platelets only stick to injured areas and not like all over inside of your vessels. So the platelet pseudopods, and if you remember back to cells, pseudopods are like arm-like projections on cells that can grab onto things. They stick to the damaged vessel walls and other platelets. So once that vessel is cut and that prostacyclin is not there anymore, the pseudopods of platelets will grab onto that damaged area and they'll hold it, and then they'll start attaching to other platelets around it, eventually causing a plug, a whole bunch of platelets stuck together in the injured area. The pseudopods contract, pulling each other tight, which creates that plug, right? The platelets are going to degranulate and release a variety of substances. So they're going to release serotonin, which is a vasoconstrictor, so we're going to further constrict that blood vessel. ADP, which attracts more platelets, so just keep bringing the platelets in. And then thromboxane A, okay, which promotes platelet aggranation, degranulation, and vasoconstriction. So it's basically another platelet attractant that helps it pull together and hold that plug. Okay, this is a positive feedback cycle until the vessel's break is healed. So as long as there are platelets being attached to that area, they're going to keep releasing these things, and it's going to keep attracting more and more and more and more until there is no ruptured area where the platelets are sticking. So once that prostacyclin goes back and the endothelium is repaired, it will shut off. But until then, it will continuously keep doing this as a positive feedback cycle. And the final step of hemostasis is coagulation or clotting. So this is the last and most effective defense against bleeding. So of course, the goal is to get the blood to clot, right? So the actual process of coagulation is converting plasma protein, fibrinogen, that we talked about when we talked about plasma, into fibrin, which is a thread-like framework that actually builds a clot. So fibrinogen is very fibrous, and it converts to fibrin, and it actually forms this thread-like, basically, shield clot over the injured area. In order to do this, there have to be clotting factors present. Okay, so you can have them in your plasma, you can have them produced by the liver, and then they are also released during the reaction cascade when an injury is present. So fibrinogen converts to fibrin if the clotting factors are present. And we'll talk about those clotting factors in just a second. Where do these clotting factors come from? So there's two different ways. You can have an extrinsic or an intrinsic pathway. The extrinsic is much faster. So it's initiated by the release of tissue thromboplastin from damaged tissue. So when tissue is damaged, it releases thromboplastin, which is also referred to as factor 3. As soon as this is released, it causes a chain reaction to release factor 7, 5, and 10. Okay, and those are the three clotting factors needed to initiate the clotting process. So one cut just sets off a domino effect really, really quick. This is the fastest way to get to clotting. So it's easier to clot blood if there's an injury somewhere than to just make blood clot without any damage. So the slower pathway is the intrinsic pathway, and this means that there is no damaged tissue, and it's initiated by the platelets inside of the body. So it's initiated by the platelets releasing factor 12. Okay, and then this causes this cascade event to factor 11, factor 9, factor 8, factor 10. And you have to go all through these factors, and then it releases the prothrombin that is released when tissues are cut. And then it goes to thrombin and fibrin, and then you have a clot. So when you have a cut, you skip all of these factors and you go right to the prothrombin instead of having to do all the factors above it. But the intrinsic pathway is much slower because there's not an injury. So I'm not expecting you to remember all of these factors. You just need to know that fibrinogen converts to fibrin when clotting factors are present, and that the intrinsic pathway is much, much slower. If you get a cut, you clot way faster than if your body's trying to clot internally without an injury. In order to complete the coagulation process, you have to have the activation of factor X or factor 10. Okay, this leads to production of prothrombin. And remember, you can get prothrombin activated if you just get cut. Okay, so either you have the activation of factor X or prothrombin is activated. That converts prothrombin to thrombin, which then converts the fibrinogen into fibrin. Okay, so this is how blood clots. It is a positive feedback mechanism. As long as you continue to have prothrombin being released, it will continue to convert it and convert the fibrinogen into fibrin. Once there is no more injured area, or you're not activating that factor X anymore, you will stop doing the clotting process. All right, so what happens to blood clots? So when blood clots, it retracts within 30 minutes. It doesn't just stay clotted in your body forever, because that's going to cause massive problems, okay? So platelets have a derived growth factor that is secreted by platelets in endothelial cells. So this is a mitotic stimulant that makes your body fix muscle in blood vessels and tissue really quickly. So if you get cut, platelets sense an injury, they're going to release these growth factors that cause your body to do mitosis really quick so it can heal itself. Okay. And there's fibrinolysis, which is dissolution of a clot. So after you have done this mitosis and healed whatever injury you have, the clot is going to get dissolved. Okay, so factor 12 is going to speed up formation of a specific enzyme that's going to convert everything into a clot dissolving enzyme that breaks up the clot. I'm not going to expect you to remember the names of it. Just know that when clots are done, clotting injuries, they dissolve, because we don't just need 1,000 clots floating around in our body. Just asking for trouble with that. So the platelets make the body do mitosis really quickly by releasing that growth factor. And then the clots are dissolved after they are done plugging up the bleed. How do we prevent inappropriate clotting? It's well known that blood clots can be a problem, right? And there are people that have blood that just clots when it should not. Okay, so in order to prevent this, we can basically repel platelets. So we can keep platelets from sticking to areas that they should not. So our body has a natural platelet repellent of prostacyclin. But what about somebody with a stent? You get a stent put in your artery because it's narrowed, it's clogged, right? It's blocked. You have atherosclerosis or whatever. You have all this buildup in there, and the artery is not open anymore. So we put a stent in there. The stent is not made of endothelial tissue. It's synthetic, and it doesn't have prostacyclin in it. So we have to take medications that put that in there, and it keeps the platelets from sticking. So repelling platelets from sticking to things is really big on clot prevention. You can dilute thrombin, which keeps you from clotting. So it slows down that clotting process. You can take anticoagulants, which slow down the clotting process as well. So things like heparin interferes with prothrombin activator, so it slows down that clotting process. And then there's something called antithrombin that actually deactivates thrombin before it can convert fibrinogen into fibrin. So these are natural anticoagulants. We also have medications that can do these things too chemically in the body. And then we have medications that can thin the blood to make it harder to clot as well.
So some clotting disorders. Clotting disorders are anything that causes the body an inability or a deficiency in clotting. So either you don't clot or you clot really, really slowly. The most common clotting disorder is hemophilia. And hemophilia is a sex-linked recessive genetic disorder. It is carried on the X chromosome and it affects boys. So thinking back to genetics, if you think about chromosomes, females have 2 X's and males have an X and a Y. If a male gets a bad X or an X with hemophilia, they don't have a second X to override it, so they end up with the hemophilia. A female has 2 X's, so if they get one X that has hemophilia and the other one doesn't, they don't have hemophilia, they just carry the trait.
Other clotting disorders include thrombosis, which is the presence of a clot in an unbroken vessel. These are really, really common in the legs of elderly people who don't walk or obese people. And they get lodged in like the calf area or the shin area, and it can cause a lot of problems with circulation in the area. A pulmonary embolism is when a clot breaks free from a vein and travels to the lungs. Okay? Anything that travels in the blood and blocks blood vessels is an embolus. So a pulmonary embolism is just the state of having one of those in the lungs. These can be fatal. And then there's an infarction, which is tissue death. And this can be caused if clots block blood supply to an organ, mainly the heart and the brain. So this is either a heart attack or a stroke. 650,000 Americans die annually from thromboembolism, which is a traveling blood clot. Typically, they travel to the heart, cause a heart attack, massive heart attack, and it can kill people pretty quick, or the brain. And if it gets to the brain and it doesn't kill the person, it usually causes severe damage. It's a stroke, and whatever side of the brain it gets on, it can cause paralysis to the opposite side of the body. So traveling blood clots are definitely not good. If anybody is known to have a history of clotting or be at risk for clotting, like having conditions like AFib, which we'll talk about in a couple weeks, they should be on blood thinners, and they should be getting their blood thickness monitored routinely because blood clots can be fatal. So when we clinically manage blood clots, the whole goal is to prevent them entirely, and then if there are any clots present, get rid of them because we do not want them traveling around in the body. So you can prevent clots by using blood thinners or anticoagulants, right? So vitamin K is the vitamin that starts this whole clotting factor. If you use medications that are antagonists to vitamin K or go against vitamin K, it will keep your blood thin and slow down the clotting process. Common medications for this are Coumadin, which other name is warfarin. That's the most common blood thinner. These keep the clotting factors such as 2, 7, 9, and 10 from activating because they all require vitamin K. So by taking these, you slow down how fast blood clots. When you're on Coumadin, your clotting time is about 2 to 3 times longer than a person that's not on it. So the normal clotting time is an INR of 1. A person on Coumadin should be between 2 and 3, so 2 to 3 times longer. And by 2 to 3 times longer, I don't mean 2 to 3 minutes longer. It's usually like 30, 45 seconds longer. So it's not enough to allow them to bleed everywhere. They are more at risk for bleeding because their blood is thin, but it's enough time to prevent the clotting. And then there's also other anticoagulants that we use, such as Eliquis and heparin. There's a lot of anticoagulant medications that can help thin blood, and they all just go against that vitamin K so it doesn't initiate your clotting factors. If you are on blood thinners, you have to be routinely monitored. Your blood thinning, the blood thickness, I guess is the word. You have to check your blood thickness every week to 2 weeks just to make sure, because it can easily get out of control and then you're at huge bleeding risk. So what happens if you already have clots? We can't prevent them because they're already there, right? So they can give you something called streptokinase, which is an enzyme. It's actually made from bacteria that dissolves clots, specifically in coronary vessels. And I know we haven't talked about the heart yet, but coronary vessels are the blood vessels that supply the heart with oxygen and nutrients. So they are imperative to life. If your coronary vessels get blocked, you probably have a heart attack coming. So we need to keep clots out of those vessels. They are really important. So streptokinase can be given to you to break down clots, specifically in that area. You can use TPA, which works faster and is more specific. It's also made from bacteria. And then you can use Hammentin, which is actually derived from a leech. So all of these things can be used to break clots that are already formed. They can also go in and break them up with like a cardiac catheterization. They can go in and actually physically break it up. They can put a filter in your inferior vena cava, which is the bottom entry vessel of your heart. And we'll talk about that. They can put this spiky pitchfork looking metal thing in there. So when the clots hit it, the spikes bust it apart. And when we talk about the heart, I'll show you a picture of that. But they can break them up in these different ways. So if we can't prevent them from forming because they're already there, these are methods that we can use to actually break up the ones that are in there. And hopefully they won't cause problems if they're dissolved quick enough.